Plastic is everywhere. It's in the food we eat, the water we drink, and the air we breathe. So what does this mean for human health? Well, to date there's been a lack of scientific evidence on the effects of ingesting plastic on human health. And only a few studies have attempted to estimate actually how much we are ingesting. My research will attempt to contribute to the knowledge advancement in these areas. My name is Lexi Crouch, and the title of my research is The Occurrence and Dietary Risks of Microplastic Ingestion by Marine Bivalves Destined for Human Consumption. Today I'll provide an overview on background information to my research study and the motivations for my research. I'll discuss the purpose of my research project and delve into the specific objectives I'll try and achieve. I will discuss the proposed methodology to fulfill these research objectives, and finally I'll give an overview of the outcomes and deliverables from my research and the potential contributions that I envision these will have. So when plastic enters the ocean, it doesn't break down, it breaks up into smaller and smaller plastic pieces called microplastics. Microplastics are commonly defined as particles less than 5 millimeters in size. They can occur as a result of degradation of larger macroplastic items, such as a plastic bottle, or deliberate manufacture, such as the little abrasive plastic spheres that you find in a facial scrub. Microplastics have been found in the surface of the ocean, the water column, and in the ocean sediments on the ocean floor. And multiple research studies have found marine organisms to ingest microplastics, including fish, crustaceans, and bivalves that are commonly consumed by humans. So marine bivalve mollusk species, including species of mussel, oyster, clams, and scallops, are particularly vulnerable to microplastic ingestion due to their habitat and their filter feeding strategy. So microplastic ingestion has been shown to harm bivalves and other marine organisms in two main ways. The first is physically. So microplastics have been shown to translocate from the gastrointestinal tract to the lymph system and circulatory system, causing effects such as cell damage, inflammation, and altering their energy expenditure. And the second impact is chemical contamination. So microplastics have been demonstrated to act as a vector for the toxic chemicals that are in plastics, but also the chemicals that adsorb to the surface of plastics from the surrounding ambient seawater. So many of the chemicals found within and that have adsorbed to the surface are known carcinogens, they're endocrine disruptors and mutagens. And studies have shown these toxins and particles can transfer to higher trophic levels up the food chain, where they bioaccumulate and biomagnify in the tissues of these organisms. And of course, at the top of the food chains is us, humans. So they pose a particular concern for human health. So marine bivalve species are commercially important species. They're valued for their meat, for their shells, and their pearls for oysters. But they also have ecological significance. They provide important ecosystem services such as carbon sequestration, nutrient cycling, and coastal protection. So bivalves pose particular concern for human microplastic exposure because they are consumed whole. So the gastrointestinal tract, or their gut, is still intact, unlike fish where it's removed. So this graph gives a good overview of how microplastics go from the ocean to our plate. So essentially, bivalve species absorb microplastics from the or when they're filter feeding from the ambient seawater. Some of these are ejected as pseudofeces or fecal pellets, but others remain in the gastrointestinal tract where they can translocate and remain inside the organism. And when they're harvested through aquaculture, they eventually find their way to our plate where they're consumed whole. So this is how it's making their way to our diet, and there are particular concerns about what this means for human health and what the risks are. So the purpose of my research study is to investigate microplastic uptake by edible marine bivalves and the subsequent dietary exposure to shellfish consumers in different regions across the globe. So this research problem was actually born out of a research project currently being undertaken by the University of Newcastle's microplastic research team, which I'm a part of. So we've identified that a couple of knowledge gaps, including the geographic influence on microplastic uptake and the dietary exposure to humans, specifically from bivalves. So these are some research gaps that my research project is going to try and address. So there are 3 research objectives in my study. The first is to estimate bivalve ingestion rates in different regions, and I want to do this at the present and also extrapolate for the year 2100 based on the increased presence of microplastics that are projected. The second research objective is to understand the potential human dietary exposure to microplastics from bivalve consumption. Again, now and in 2100. So essentially, how many microplastics are we ingesting based on an average seafood diet? And the third research objective is to synthesize the research on human health impacts of microplastic ingestion. So looking at what the literature is saying about what these health impacts are. Just quickly on the scope of my research. So I'm only looking at bivalve species that are consumed by humans. So those species that aren't consumed by humans are excluded. Freshwater and deposit feeding bivalves are also excluded from my research because less published literature is available on these species. So my focus is really on those filter feeding extant marine bivalves that are commonly consumed by humans across the world. Also, the study will not conduct field work or lab experiments. My focus is on conducting empirical research and conducting a desktop research project using information that's available in the peer-reviewed literature and robust extrapolations, estimations, and statistical analyses. And you can find more details on the technical aspects in my research proposal. So now moving on to the methodology. So my research study was broken down into 4 stages. The first stage is the development of a database. So I'll systematically review the literature and develop a database of microplastic loads in bivalves in different regions in the world. So many studies have used different methodologies to identify and quantify these microplastic loads. And my research proposal outlines how I will address this issue to ensure the data that I collect is comparable and as accurate as possible. You can refer to my research proposal for technical aspects on how that will be done. So the second stage is looking at estimating bivalve microplastic concentrations, because my literature review identified that there are many studies on marine microplastics occurring in bivalves is missing for certain regions. For example, Australia, New Zealand, much of America, Russia, and the Middle East. So we haven't got data on microplastic loads in bivalves for these regions. So what I propose to do is to estimate it. So essentially what I'll do is if we know how much microplastics are in the water in a certain region, and we know how much microplastics are ingested by bivalves on average, then we can estimate the average microplastic loads in bivalves for that region. So the data that we'll use, you can see here some of the variables, and for more information you can refer to my research proposal on technical details on how these estimations and extrapolations will be completed. So we'll do this now and also for 2100 to look at what those concentrations and loads will be based on what the projections are for microplastics in the ocean in 2100, because it's expected to increase a lot. The third stage in the methodology is using the results from stage 1 and 2, as well as average shellfish consumption data, to estimate how many microplastics people who eat bivalves are ingesting and how this differs by each region. So only 3 studies have estimated this dietary exposure through bivalves. And my study will use the same methodology as these studies. And these are the studies here. So there's one being conducted in Europe, Tunisia, and Korea as well. I'll also use statistical analysis, namely regression modelling, to predict what these ingestion rates will be in 2100 based on average shellfish consumption trend data. Again, you can refer to my research proposal for more information on the data sources and the methodology and technical details for these calculations and estimations. And I will be mapping these results using geospatial mapping, so heat mapping, so we can easily visualise how the results differ by region as well. The fourth and final stage will be synthesising the results, interpreting them and discussing the results in the context of the latest peer-reviewed literature as part of my dissertation. So there are 3 key outcomes that are expected from the research. The first is a database, a robust database containing information on microplastic loads in bivalves by region, as well as what the dietary exposure is, both for present and for 2100. So essentially, how many microplastics is an average person consuming per annum in each different region based on the available data. The second outcome is geospatial mapping, as I mentioned before. So this makes it really easy to compare results and interpret and analyse those results as well. So this will be done for bivalve ingestion loads and dietary exposure. And of course, the third deliverable will be my dissertation, which will include full literature review, methodology and assumptions, the results from the 3 stages of research, and discussion, interpretation of those results, conclusions and comments on future research that is needed. So the outcome from this research will contribute to the current knowledge gaps on microplastic occurrence and human health risks. So by understanding dietary exposure, scientists will be able to investigate the impacts of different ingestion rates of microplastics on human health. And by identifying where geographically those risks are greatest, the hope is that this research will inform geographically targeted strategies to mitigate microplastic contamination in the ocean and manage the potential human health risks. So in conclusion, we know that bivalves are considered a key pathway for microplastics to transfer to humans because they're consumed whole, unlike fish. However, at present, scientists do not fully understand the risk that microplastics pose to human health when consumed via seafood. So this research study will provide estimates of microplastic ingestion by bivalves and estimations of dietary exposure to humans. And the hope is that this will assist in identifying areas that are geographic areas that are at greatest risk of exposure. And this will contribute to knowledge advancement and will assist researchers in understanding the risks to human health. And the goal is to inform mitigation and management strategies. Thank you very much.